Welcome to this video of microbiology in which we are going to see the structure of bacterial cell. Bacterial cell is a prokaryotic cell, that means a cell which is lacking in well-defined nucleus. It is not having nuclear membrane which is separating the nucleoplasma from the cytoplasma of the cell. The membrane bound organelles, they are also absent in case of bacterial cell. We will start the structures from the outermost part of the cell. In some bacteria, you have the outermost covering as a capsule. Capsule is made up of exopolysaccharide in majority of the bacteria. The function of capsule is it protects the cells from desiccation in soil. It prevents the phagocytosis of the pathogenic cells, and it also helps in attachment of the cell. Inside the capsule you have cell wall which is made up of peptidoglycan. There are two types of cell walls, gram positive and gram negative. The gram positive bacteria, they have thick layer of peptidoglycan, and the gram negative bacteria, they have thin layer of peptidoglycan which is also surrounded by lipopolysaccharide layer. In short, it is called as LPS. LPS layer is also called as outermost membrane because it is having nature similar to lipids. Below cell wall you have cell membrane. The function of cell wall is it protects the cells from osmotic shocks. If the cells are present in hypotonic conditions, the cell wall, which is rigid and hard structure covering the cell, it protects the entry of the water inside the cells, and thus it prevents the cells from osmotic shocks. Inside the cell wall you have cell membrane which is made up of lipid bilayer. The outer covering of this lipid bilayer is made up of heads of the phospholipids, and the inner part of this lipid bilayer is made up of tails which are hydrophobic in nature. The cell membrane is involved in transport of many ions and molecules inside and outside of the cell. Inner to the cell membrane you have cytoplasm of the cell, which is viscous liquid present in the cell. This viscous liquid contains different types of nutrients molecules such as carbohydrates, proteins, amino acids and lipids. Along with these molecules, it has different types of enzymes which are involved in metabolic activities and different types of intermediates of the pathways. In the cytoplasma you have very important molecule called as DNA molecule. It is also called as chromosomal DNA of the cell. This molecule is circular in nature. The function of DNA is it is having all the genes which are required for metabolism of the cells, its reproduction and its survival under different conditions. The DNA is made up of deoxyribonucleic acids. Along with the main DNA of the bacterial cell, you have small DNA molecules which are circular in nature called as plasmid DNA molecules. The plasmid DNA is a circular as mentioned earlier, and it is carrying some genes which are important for survival of the cells under particular conditions. The genes such as resistant to different antibiotics, resistant to heavy metals, they are present on the plasmid molecules. The DNA, genomic DNA of the cell, it is attached to the mesosomes, which is nothing but a infolding of the cell membrane. In the cytoplasma, you have ribosomes, which are the sites for protein synthesis. In bacteria, ribosomes, they are made up of two subunits, larger and smaller subunit, and the total size of the ribosome is 70S. S stands for Swedberg unit. Along with these main structures, on the surface of the bacteria you have small extensions called as fimbriae or pili. These structures helps in binding of the cells to different surfaces. The pili, they are made up of protein pillin. These fimbriae, they are also responsible for transfer of genetic material from one cell to other cell during the process of conjugation. Some of the bacteria, they have large extensions called as flagella. This flagella is responsible for motility of the cell. The flagella utilizes energy of proton motive force at the membrane, which helps the rotational movement of the flagella. This rotational movement of the flagella pushes the cells in the viscous medium. Flagella is made up of three parts: basal ring structure, then hook, and then the filament, which is a long extension on the cell surface. So in this way we had seen different structures of the bacterial cell and their functions in short. Thank you.